Bismillahirrahmanirrahim. Today's topic is very important from biochemistry point of view. There are two pages of first aid, 74 and 75. These are very important. On 74, there are multiple reactions, and on 75, I have jotted down important MCQs related to nutritional energies, like each compound and requirement of each major nutrient, how much energy it will yield. Uh, Most of them are not given in books. So coming on to 74 page, there are multiple reactions here. We need not to be worried about them. They are all interlinked and they are very basic uh, chemical reactions of our body. For example, we know that we have three things. First is glucose, that is a carb. The second is protein, here down, protein metabolism, and the third is lipid metabolism. So everything is related to each other at certain levels. Most of the reaction here is breakdown of glucose, that is called glycolysis. Glucose is 6 carbon compound, we know, and it breaks down to the level of pyruvate, that is 3 carbon compounds. So one glucose will create 2 pyruvate, that is an MCQ. And pyruvate is in fact an intermediate molecule, because pyruvate will enter the Krebs cycle or TCA cycle, tricarboxylic acid cycle, because pyruvate, we know that it has 3 carbon, so the acid is tricarboxylic acid, most of these 3 carbon chemicals, so it is TCA cycle. But through another chemical conversion, that is acetyl CoA formation, so pyruvate will convert into acetyl CoA through pyruvate dehydrogenase, and this is in mitochondria now. Pyruvate enters the mitochondria. We know mitochondria are the powerhouses of cell, and before pyruvate glycolysis is all this reaction is in the cytosol. So glycolysis is cytosolic reaction. For example, RBCs do not have the mitochondria, so it will be the glycolysis that will be main source of energy. But most of our body cells have mitochondria. So first glycolysis takes place, then the Krebs cycle takes place, the whole glucose molecule is broken down and energy is released. If we learn that how much ATP it creates in the end. First, we need to understand that some of the MCQs are coming from glucose to pyruvate conversion and then pyruvate to acetyl CoA conversion. That two pyruvate intermediates from one glucose are formed. So pyruvate is an intermediate between glycolysis and Krebs cycle. Then, if we know this is the alanine is an amino acid, and protein enters into gluconeogenisis through this mechanism, that the alanine amino acids, they convert to pyruvate and it can move back through this line to glucose, and then glucose can move to this direction or body glucose level will be maintained. This is how protein enters into the terminal step of glycolysis through alanine. And about lipid metabolism, lipid enters through this pyruvate and related chemicals. This enters a step down into acetyl CoA, and these two reactions are important. The protein enters through alanine and fatty acids, we can see here. This reaction is not given here, but fatty acids move through acetyl CoA. So lipid metabolism enters this glucose metabolism through acetyl CoA. We know acetyl CoA is in the mitochondria now, because this TCA cycle is taking place in mitochondria. So this pyruvate dehydrogenase enzyme at position 14 converts pyruvate to acetyl CoA, and this is happening in mitochondria. Above pyruvate, everything happening in the glycolysis in cytosol. And this is aerobic glycolysis. Unless pyruvate converts into lactate, this would be anaerobic glycolysis. And some other few reactions of HMP shunt, fructose metabolism, ketogenesis is here, and urea cycle is here. They are all linked together. Gluconeogenisis is opposite of glycolysis, and glycogen storage of glucose. Let's make a story that someone is eating too much of glucose, and that glucose in the blood will be converted into glucose 6 phosphate. Phosphate form will convert into glucose 1 phosphate. uh, of, so this one glucose 1 phosphate will be converted into glycogen form ultimately. So glycogen storage in body will increase, and glycogen is stored in mostly liver and muscles. And whenever there is not much insulin in the body, for example between meals, here liver activates glycogen phosphorylase in insulin deficiency. This is an MCQ between meals. So here is this enzyme. Glycogen will be converted into glucose 1 phosphate, that will be going to glucose 6 phosphate, and ultimately glucose level will be increasing from glycogen. More glucose will be produced between meals because of risk of hypoglycemia increases between meals. This reaction is important, and other mediator of this reaction of to increase the glucose level between meals is adrenaline and glucagon. So insulin deficiency or glucagon excess, we know glucagon increase blood glucose level. So first it will be by increasing this glycogen phosphorylase enzyme, deficiency of insulin, or excess of glucagon, that is opposite hormone, or excess of adrenaline. We know the stress hormone adrenaline or epinephrine causes hyperglycemia, because here it will increase glycogen conversion to glucose 1 phosphate, and this is phosphate form is converted into glucose 6 phosphate. So one position phosphate will move to 6 position by phospho of fructomutase. So this PFM enzyme is important because it will lead to the one form to mutase enzyme it to the other form at the sixth position. It doesn't add phosphate, just take it from one position to 6. And another question is asked that which step of glycolysis is reversible. In fact, there are 3 steps: the third, 7th and 13 here, glucose to glucose 6 phosphate conversion, or sometimes it is given as glucose to fructose 6 phosphate directly. So we can say the third and the next step. The 7th step is also irreversible: fructose 6 phosphate to fructose 1,6 bis phosphate. So this is irreversible because we know that once glucose is in the phosphate form, it the phosphate form will convert into fructose 6 phosphate, and fructose is a different chemical compound. So glucose do not move back unless there are enzymes for gluconeogenisis. That is a separate reaction just shown here to understand that both are opposite to each other. But irreversible step of glycolysis: glucose to fructose 6 phosphate and fructose 6 phosphate to 1,6 phosphate, and the last 13th one, PEP conversion into pyruvate. But only this first one or second are asked. So another very important thing to understand that of gluconeogenesis, that formation of new glucose from either protein or through alanine path, alanine, and this reverse of glycolysis pathway. um, this G6P glucose 6 phosphatase enzyme at position number, we can say here it is at position number 4, glucose 6 phosphatase enzyme. So this is the reaction in opposite direction of first step of glycolysis, that is conversion of glucose 6 phosphate into glucose molecule. So this will form the new glucose molecule independent of any phosphate or any other compound. If this enzyme is deficient, glucose 6 phosphatase, then it will lead to von Gerke disease. This is important MCQ. Why? Uh, it has certain symptoms, because if glucose this reaction is not taking place, We know that this is the last reaction of gluconeogenisis. So glucose level won't be raised. New glucose will be trapped somewhere down, like glucose 6 phosphate form or fructose forms, all behind this. Even if body has too much of alanine or pyruvate available. So G6 uh, P glucose 6 phosphatase, I have written it as G6P, otherwise it is glucose 6 phosphatase, is lacked in muscles. It is not present in muscles, so muscles cannot form new glucose. So gluconeogenesis is not taking place in muscles, rather in liver. This is another MCQ. This glycolysis is taking place in every cell, but this gluconeogenesis is not taking place in muscles, that is the most of the body part, because it lacks this last step of glucose formation in gluconeogenesis, or first step, or opposite of first step of glycolysis, we can say, mediated by glucose 6 phosphatase, the same enzyme which is important in von Gerke disease. So we'll link gluconeogenesis and von Gerke here. Glucose 6 phosphate lacked in the muscles, so muscles cannot participate in gluconeogenesis, one MCQ. And hypoglycemia resistant to glucagon in von Gerke disease is seen. How? If someone is having von Gerke disease, he or she will lack this glucose 6 phosphatase at fourth position, this reaction. So even if these glycogen storages or this pyruvate and alanine available in body, new glucose cannot be formed from this reaction, because it will be reaching maximum to glucose 6 phosphate, and if glucose 6 phosphatase enzyme is deficient, terminal independent glucose molecules cannot be formed. So if a patient is given glucagon in excess to break down this glycogen from here by activating glycogen phosphorylase, or activates gluconeogenesis enzymes, multiple enzymes are activated by glucagon as well, because it will increase the level of glucose opposite to insulin, but it will reach at this point but won't move up to glucose level, because this glucose 6 phosphatase is deficient. So patient will be having resistant hypoglycemia, resistant to glucagon treatment. So in von Gerke disease, or they can ask that for which enzyme would be deficient, which will be glucose 6 phosphatase deficiency. Even a resistant case, resistant or hypoglycemia will be there to glucagon. And then now we can see that galactose is another molecule that can enter into this pathways of glucose 1 phosphate. But the first reaction is by galactose 1 phosphate uridyl transferase, deficiency of which will cause galactosemia. This is an MCQ asked, that galactose won't be converted into galactose 1 phosphate, that enters into glycolysis through this reaction in the next step, and galactose will accumulate ultimately, that is galactosemia, severe galactosemia, deficiency of UDP, uh, deficiency of galactose 1 phosphate uridyl transferase, that you have to remember. And then a very interesting uh, thing here in the urea cycle, that we know that end product of protein is urea, and of uh, DNA basis like purine pyrimidine is uric acid. So urea and uric acid are different things. For protein it is urea, and the ammonia is produced end product of many amino acids. Amino acids have ammonia, that's why they are amino acids in the protein. So ammonia, it is combined with one aspartate, that is a very important amino acid, to make the urea. This is an MCQ asked from urea cycle. And arginine will produce nitric oxide as another reaction asked. So most of the MCQs from this page are covered. We will move on to the next page. So page 75, nutritional energies. I have jotted them down here, so we don't need to read the whole page above. They are not much given here. 1 gram of lipid or fat, it will yield the highest energy, that is 9 kilocal. Highest energy by any of the main nutritional things like lipid, protein, carbs. It is the lipid or fat, that is 9 kilocal. 1 gram of protein will yield almost half or less than half, that is 4 kilocal. That is also asked. And 1 gram of carb, again same 4 kilocal. So carb and protein same. Lipid or fat, that is high, double, more than double, 9 kilocal. Sometimes this can be asked. 1 gram alcohol, it will yield 7 kilocal, that is in between this protein, carb and lipid here. And 1 gram of after alcohol, the same energy is produced by 1 ATP as well, 7 kilocal. That is also asked, not given in the book. Then 1 NADH gives 2.5 ATPs precisely. In FSC we have read about 3 ATPs, but now precisely calculating it is around 2.5 ATPs. And 1 FADH2, basically is FADH2, it gives 1.5 ATPs. So 2.5, 1.5, and body requirement of energy is 25 to 30 kilocal per kg per day. If we multiply it with 70, so we will get like 2100 calories. That is the daily requirement of usually one person, roughly. So it is 25 to 30 kilocal. And 1 degree rise in temperature of body will raise 8 degree rise in basal metabolic rate. That is also an MCQ that should be remembered related to this topic. And then glucose metabolism, we just read that in aerobic glycolysis plus TCA cycle, it will produce total of 30 to 32 ATPs. This is an MCQ. Sometimes 30 or 32 can be given because 2 ATPs are used in glycolysis as well. So they are sometimes added, sometimes not. Glycolysis 6 ATP. Link reaction of we know the pyruvate to acetyl CoA, and then TCA itself 20 ATPs. These are not important, but overall 30 to 32 are important. Then anaerobic metabolism produces 2 ATPs, that produces lactic acid, we know, just 2 ATPs, anaerobic glycolysis, and 30 to 32 and just 2. And alactic oxygen depth would be added later. It is 3.5 liters. You just have to remember it. It is under the curve certain liter of oxygen required for alactic oxygen depth to compensate it. This is 3.5 liters. So 30 to 32 aerobic, 2 ATP anaerobic, and 3.5 liters is oxygen depth for alactic oxygen depth. The protein metabolism related question. Proteins required for daily are 1 gram per kg of body weight, so roughly 70 gram for one person. And daily protein calories should be 15 to 20% of the diet. Maximum is of the carbs in 40s, then of the lipids in 30s, and then of this protein. That is the percentage of total calories. It should be 15 to 20% roughly. Here it is a question related to protein is 1 gram of protein will yield 4 kilocal. Here it is daily protein requirement 1 gram per kg of body weight, and daily protein calories is a different thing, overall percentage of calories of the diet. So we have to make a difference between them and remember them as such. Hope this made sense, and these are the MCQs that will be asked in exam. For most of the part you can read in details, but this is how these reactions are jotted down together and should be remembered. Thank you.